Good day, this is Professor Will, CBMD PhD. Today's Wednesday, December 22nd, 2021. It is 3:22 and 14 seconds, a.m. Eastern Standard Time. Today's research, neuroblastoma. So let's jump to it here. A study on genetic changes in neuroblastoma. Date December 10, 2021. Source, University of Berlin. When cells within a single tumor differ in terms of their genetic makeup, this is referred to as intratumor heterogeneity. Researchers have been able to reconstruct the process by which the genetic heterogeneity develops in neuroblastoma, a type of cancer which primarily affects young children. According to their findings, the genetic makeup of individual tumors show marked spatial and temporal variability. In the study, researchers were able to show that the genetic changes typically associated with neuroblastoma can both disappear and emerge over the course of the disease. And these mutations are not evenly distributed throughout the tumor. Rather, they occur in distinct sections, or even in individual cells, giving the tumor a mosaic-like appearance. Cancer is driven by evolutionary processes. Cancer cells undergo constant genetic change. They are engaged in a fight for survival, including against other cancer cells. Each cancer has its own phylogenetic tree, which depicts the way in which a tumor evolves. Some cancer cells become metastatic, while others develop treatment resistance. The researchers analyzed a total of 140 neuroblastoma samples. The biopsy samples were collected from 10 pediatric patients at various points during the clinical course and covered multiple tumor regions. And sample analysis involved in the use of multiple modern sequencing techniques, followed by computer-assisted evaluation. The researchers focused their analysis on the neuroblastoma-associated genes ALK, MYCN, and FGFR1, which play an important role in both clinical course and treatment. According to their results, changes in the ALK and MYCN genes were not present continuously throughout the course of the disease, nor were they found in all tumor cells. The changes in the ALK and FGFR1 genes can offer useful treatment targets, particularly in relapsed patients. The researchers found that, in some patients, ALK mutations, which were present at the time of diagnosis, had disappeared by the time the tumor was surgically removed. Furthermore, changes in the FGFR1 gene were only found in distinct tumor regions. The researchers were also able to identify an instability in the number of gene copies present in neuroblastoma cells. In some cases, cancer cell clones showed early divergence from the primary tumor, breaking away to infiltrate other organs where they formed metastasis. The process of recording detailed spatial and temporal changes in the copy numbers of specific genes is extremely complex. Researchers developed an algorithm capable of producing a precise reconstruction of these copy numbers and used this method to produce evidence of continuous structural evolution in a range of different cancers. Researchers have now been able to extend this to neuroblastoma, where they were able to show in detail how the cancer genome undergoes structural changes. Researchers are now in a better position to understand how neuroblastoma cells behave, and this knowledge is essential in relation to patients who suffer a reoccurrence of their disease because their treatment often requires the use of personalized and targeted therapies. When a tumor presents as genetically heterogeneous, targeted molecular therapy may well capture a majority of the abnormal tissue, but, crucially, will not capture all the affected cells. The cancer will then be able to regrow from those remaining cells. The research findings have less relevance for neuroblastoma diagnosis and therapy selection, and this is because diagnosis is already reliable thanks to technologies that have undergone decades of testing, such as imaging, urine testing, and single tissue biopsies, and because chemotherapy, targeting fast-growing cells, remains the treatment, a choice for the primary tumor. However, if the disease recurs, following treatment, targeted treatment options become particularly important. The treatment selection, which is based on a single tissue biopsy from a single tumor region, is unlikely to adequately address the tumor's genetic heterogeneity. And when a relapse occurs, they should therefore consider using state-of-the-art sequencing techniques to analyze tumor tissue samples harvested from multiple regions. And this would provide researchers with as much detail as possible about the disease, enabling researchers to further improve the decision-making process pertaining to the selection of personalized therapies. The researchers are now testing other methods in hope of resolving some of the technical challenges which remain, and these include the use of single-cell-based technologies and liquid biopsies, a new type of blood test which analyzes genetic information shed by tumors into the bloodstream, and using the analysis of multiple blood samples over the course of disease. And these techniques will provide evidence of any genetic changes taking place without the need for invasive surgical biopsies. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.